position is a non-zero element. So your eye is fixing at a certain point that's not straight ahead. And they found that this is related to, linearly related to the amplitude of the eye movement, with this k constant equal to 5 experimentally derived. And then the next step was to see how the firing rate changed as a function of the the velocity of eye movement. And so they had a person move their eyes across the screen and just measure the firing rate at that 0 point in the middle. So the E is going to be 0, but the velocity of eye movement is non-zero. So they found that this is also this is linearly related to the velocity at that 0 point. And so by combining these three experiments, the final force in relationship, or the final firing rate in relationship to the eye amplitude of movement is this basal R0 plus this linear relationship between eye movement, and then also the velocity of the eye movement. So in kind of relating the relationship between firing rate and also the force as a function of the eye movement, both of these are of the same form. And so we can see that the relationship between force and the firing rate of the motor neurons is linear. So the next step was to see actually what command was required to generate a firing rate to allow a saccade to actually happen. And so in the lecture, Dr. Cullen derived that if you actually work out just applying a simple step command to this equation, the time constant is too slow to make a saccade. So the question here is how do people make saccades if you can't just apply a step command and make it happen? So the solution is to apply a pulse in combination with a step. So they found that you needed an extra burst to actually generate the command to complete a saccade, and then the step is needed to actually fix your eye at a certain point. So the pulse occurs in these burst neurons, so that's indicated by the red, and it just happens in the beginning portion of the saccade, and then the step is initiated to hold the eye at the new position, and this is accomplished by an integrator in the nucleus prepositus, and then it holds your eye in this new position. So to actually create a step function, you require a neural integrator. And so this is modeled by just two neurons kind of in a circular connection point. And so these burst neurons are connected to the neural integrator, which then synapse on the motor neurons. And then the step function is applied in this connection right here, and then the impulse is applied through the burst neurons. And then one key point here is that without each of the two systems, you can't perform a saccade. So without the burst, obviously the saccade is too slow. And without the integrator, or even if the integrator is leaky, the eye drifts from its desired position, and so you can't hold a position where you want to. Mm-hmm.
So I guess this is similar to what Professor Zhang was saying before with fixation neurons and burst neurons. But before, on the first slides, when we were talking about which parts of the brainstem were controlling which muscles to use, mm-hmm. Are these two different types of neurons both going into the brainstem then telling the brainstem how to move the eye, or are these two different parts of the brain that are all that are directly moving the eye?
So these both synapse on the motor neurons, in the same motor neuron.
Oh, okay.
So the indicated right here is, the motor neuron that both of these interact with themselves.
And in the slides before, what were those groups then?
Are you talking about this?
Right. What is so the burst neurons and fixation neurons synapse to these three groups right there, and then these motor neurons are what all the muscles?
Yes.
Yeah, okay.
Yes. And then these motor neurons are each involved in different types of eye movements, but they control the actual muscles themselves. Okay. Any other questions? Okay, so when actually performing a saccade, here's kind of the breakdown of the pathway. So first you have an excitatory input that comes from the superior colliculus, and this is divided into the caudal and rostral areas of the colliculus. So the burst neurons are excited from the caudal area. And so the saccadic burst drive derives from this portion. And then the next step is that the burst neurons drive it through that impulse function, but they also drive it through the integrator function via the prepositus. So both are connected to the motor neurons themselves. And then the caudal superior colliculus during this saccadic burst sends inhibitory projections to both the rostral superior colliculus and the pause neurons, which makes it so that you're not fixating during this burst activity. So then once the burst ends and you're onto your fixated point, the burst neurons turn off, and then this inhibition of the pause neurons and the rostral superior colliculus ends, and so you're able to fixate on a specific point in space. And this is all, you can see the activity of each of these neurons in this diagram during the eye movement and then during fixation. So this is going into Dr. Cullen's second lecture, which is focused on the vestibular system and then also the VOR reflex. And so first, just to start off, in the vestibular system, there are 5 end organs, and they basically sense where you are in space and the motion dimension of your movement. So first, the first thing that is sensed is linear acceleration, and this is accomplished via the saccule and utricle. And also the otoliths, which are calcium carbonate structures. And the angular acceleration is sensed through the semicircular canals, which are fluid-filled and basically sense different types of angular acceleration. Mm-hmm.
So the otoliths are a sub-part of the utricle and saccules? Or inside of it?
Yes, yeah.
And what's the structure of? It's made of calcium carbonate, but what is the structure?
So it's made of a calcium carbonate structure.
Yeah, but what is the structure?
In terms of its shape, or?
I mean, is it the part, like most of it, is the calcium carbonate? Or?
Yeah, it's like composed of these little calcium carbonate stones, basically, or like little, little like formations that are inside these elements and able to sense the acceleration.
And that's functionally significant in some way, like it couldn't do its function without the otoliths?
Yes. Yep. Any other questions? Okay. So yeah, the yellow portion in this diagram is the liquid that fills the entire semicircular canals, and this potassium-rich system is what's able to sense the movement through the endolymph movement during angular acceleration. So the next kind of portion of the anatomy are these 2 types of afferents, which are irregular and regular. So type 1 are irregular, and they're involved in transient motion, and they're type 1, so they innervate type 1 hair cells. And type 2 are regular, so they're not involved in transient motion. They fire pretty much every 10 milliseconds, and they innervate type 2 hair cells. Yes.
I had a question because on the slide it says that the regular ones actually have lower gain and other kinds of properties like that. Does that have a functional or significant functional difference to it?
I'm not sure. In terms of the gain of the type.
Well, it could be related. I'm just wondering if the regular has a specific function and purpose for the development of it.
Do you know?
It says for my memoir.
We can check on that. So you're saying that type 1 or type 2 has a larger,
No,
a smaller gain? Okay.
I mean,
And asking about the,
just wondering why it was low.
okay. Yeah, we can check on that definitely if it was mentioned in lecture. So the next kind of point through the pathway is that when you have this head velocity that's applied, the firing rate through the different systems, so through the afferent and then in the vestibular nuclei in the central nervous system, the time constant becomes elongated. So the response essentially to the applied head velocity is slowed down. So you can see this through the step function that is the head velocity, and then the applied velocity, the slope of the firing rate becomes less steep during the progression through the central nervous system. And the reason why this is the case is experimentally derived through the projections in the cerebellum and the vestibular nuclei. So after they applied lesions to these two areas in the brain, they found that the response actually returned to the original 5 milliseconds, or 5 seconds versus the 21 seconds. And so the reason that this time constant is the case is due to these structures. Do these projections. Yes.
For the time constants, why, I think this question was asked on Piazza but never answered. Why are they in the magnitude of seconds? What?
Why is it so long?
Yeah, why did it take so long to decay that now?
I mean, I think intuitively, because this is like a reflex over time, like your correction to that point can be longer than the actual eye movement itself. I don't know if there's like a specific reason that you're looking for, but.
And also, what is the significance of elongating the time constant? Because I mean, we can know why it happens and what part it happens, but what's the purpose of having a 21-second time constant instead of a 5-second time constant?
I think knowing that it exists and why it exists, and just like understanding the relationship between firing rate and the actual head velocity, and how this is kind of degraded throughout the transmission of the information to the central nervous system is the main point.
Okay.
What are the spikes in the eye velocity?
The spikes.
Yeah.
So this is just like integrating the eye velocity information into a smooth function.
Right. That's the eye velocity itself that's spiking like every 2 seconds or something.
Yes. Yeah. So these are like the quick phase, and then this line here is what's actually the smooth phase of the eye velocity.
Like the smooth line is what happens when there are lesions. So when there's no lesions, there's still spikes, right?
Sorry, what is your question?
There's 2 lines to the eye velocity. The smooth one is the one where you have lesions in the nodulus, and it goes down to 5 seconds. So in the normal case, when there's no lesions, it's like this and it spikes, and it goes and keeps spiking. I'm not sure why the eye would suddenly shift back and forth.
So they're not. It's not. One is the smooth phase and one is the quick phase. It's not representing the difference in.
Magnetic velocity, right?
Yes, in smooth and quick velocity.
Okay, and then which one's the one with spikes?
The quick.
Okay, never mind.
Yes.
So I think one of the things he's saying is the one that's represented by just the line is the one that mimics the time constant where you have a lesion, which is why it decays so much quicker.
Right.
So the one without lesions has these spikes in it. So I am also confused as to the purpose of spikes.
So is the question that you just don't know what the spikes are, why the spikes are there?
So it just randomly, are they just saccades then?
So because of the lesions to these structures, the spikes are noticed in the actual experimental output.
No, those happen without the lesions is the smooth one, right?
I'm sorry, beforehand.
Okay.
Is there another explanation? Okay.
Forget about it.
Okay. Any other questions? Okay. So the next topic that was covered is how we process self-generated versus external movement. And so basically this is the concept in that when you actively move versus when when you're sensing movement that's coming from outside your body, you process these differently to compensate and then only encode what is actually applied to you versus what you're sensing due to your active movement. And so how this occurs is when you have a motor command signal, such as an active head movement, this is applied. And then this signal is sent to the effector muscle. And this is self-generated movement, so this is what you're actually, what the movement that you're going through in a normal motor command. And so this sends a signal to the sensor, which is the vestibular reafferents, because you actually initiated the motor command. And then if there's a movement that's applied to you, this is also input into the sensor. And so when you're calculating the x-afferents, so the external movement, you subtract what you generated by yourself, and then have this x-afferents, which is the external stimulation. So basically this is taking into account what movement that you initiated and then subtracting it from the output signal using this efferents copy.
Copy is sent from the muscle or from the brain?
It's sent from the brain.
So what's the vestibular reafferents? There's a line there going from the effector muscle to the sensor.
The
one that's right next to the vestibular reafferents. There's a line from the muscle to the sensor. I'm wondering if that's another thing.
So it performs this like computation using the signal from the muscle and the sensor, and then this efferents copy is sent. This is just representing the efferents copy sent to compute the output.
That's receiving input from the muscle and the brain.
So it's the actual input from the muscle is sent from the muscle to the sensor to compute the reafferents, and then the x-afferents is computed, so you actually know what the external stimulation is. So the key takeaway from this system is that it is the information is encoded differently in the afferents versus the central nervous system, the vestibular nuclei. And so in experiments in the vestibular nuclei, when passive movement and active are applied, so passive meaning that like external stimulation is applied versus active is self-generated, the firing rate is positive for this passive movement, as indicated by the bottom graph. And this is because the head in space velocity is different from the head on body velocity. So you can, when you subtract those two, you get a positive firing rate. And then in the active case, so when you're actually moving yourself, this head in space velocity is the same as the head on body velocity, because you know where you're moving. And so when you subtract these two elements, you get a 0 firing rate, because it's self-generated motion. And differently in the afferents, they encode passive and active stimulation similarly, so there's no distinction between external stimulation that's applied versus self-initiated movement. So that's all for Dr. Cullen. Are there any questions? Okay, so now I'm going to briefly review Dr. Zhang's material, and this will mostly cover the perceptron model and then some of the representations of a weight matrix in the anatomy. So first I'm going to just talk about perceptrons in a general sense. So a perceptron is applied to computational neuroscience, but it can exist outside the realm of the neural network. So the goal here is just to train a network so that it can predict an output from a given input. And that's supervised learning. So as Andy said, you know the expected output, and then you can derive the optimal weights to produce that output. So in order to do that, you use this error function, which is just taking the difference between the actual output from the model, or the actual output and then the output from the model. And the goal here is to choose weights that minimize this error function. So here you have inputs, they're summed with their applied weights, and then there's a nonlinearity to get the output. So the nonlinearity can be in terms of a sigmoidal function, a gain function. It's based on the actual model applied. So there are two methods that we went over to calculate the optimal weights. The first one is only if you have one data point at a time, and then the weight is iteratively updated so that it optimally represents the data. So this is using this gradient descent rule, where you have a constant times the partial derivative of the error function, and you can iteratively, as you get new information, update the weights that should be applied. The second way to apply the weight matrix is if you have all the data at once. So you can comprise your X matrix and your Y matrix based on the inputs and the outputs, and you can arrange them in columns, and then calculate the pseudo inverse of X because it may not have full rank, and get this weight matrix, which is X, Y times the pseudo inverse of X. So just to go more in depth on what the learning rule and how you calculate it is for a perceptron, for a single input, you can calculate the output, or for a single output, you can calculate the calculate this using all the inputs and then summing them with their weights. And this is a linear operator, so this only works for linear functions. So you have this one output for all the inputs, and then using this error function, you can derive the weights. So this is the generic equation for weights, where there's this learning rate constant, and then multiplied by the gradient descent. So the negative of the gradient descent indicates that you want something that is the most negative, so that decreases the fastest. And then when you plug in for the partial derivative here, you just take it using this equation for the error. And if you take the derivative of this with respect to y, and then you multiply it by the derivative of y with respect to w, you get the chain rule, and then you're able to do dy dw and calculate the final equation for the change in weights by subbing in for dy dw. Any questions about this? So if you change for example, this model, you should be able to derive a new weight function using the derivative, these basic properties. So this was for one single output, but for a model with multiple outputs, you do a similar computation using matrices. And so for one data point, so one output using all of these connections, you can have the summation over all of the different inputs, and then the weight applied of those inputs. So in vector matrix form, it's just this y output times the matrix of the weights, and then the vector of inputs applied. And then if you're applying multiple data points at once, you can just change this into matrix form as well, and have this Y matrix and then the weight matrix and the X matrix all multiplied together to get the final output from the initial inputs. So in order, like I described before, to do this with multiple data points, you have to derive the pseudo-inverse, which is given right here as X transpose times X, the inverse of that times X transpose again. And this is because you can't assume that this X matrix is invertible because you don't know if it's full rank. So the next topic that Dr. Zhang covered in the second midterm material is what this weight matrix actually means biologically. So this is linked to the topic of receptive fields. So in the auditory system, the weight matrix in one dimension is just the frequency. So the optimal frequency in a neuron, and then what its weight is what the connection weight is based on the neuron's preference, essentially. And so you can see here these little troughs next to the optimal weight, and this is due to lateral inhibition, because at the optimal weight, this is what is actually encoded for, and then anywhere beyond that to the direct left and right is inhibited. And then the next concept is incorporating this frequency, which you're familiar with time. And so this is the idea that over time you integrate in small windows the sounds that you hear. And this results in a kind of integration over a small point, and then you can integrate this time and have a optimal time and optimal frequency that the neuron is firing at. So the same concept can be applied to the visual system. And so the 2D representation of a receptive field for the visual system is just an XY coordinate, what that specific point prefers. And so in 2 dimensions, this is the firing rate is represented by the Z direction, and this is just an XY coordinate. And you can see, if you look closely, the same troughs next to the optimal position for that point. And this is, again, lateral inhibition of the nearby areas. And then, again, with the incorporation of the time window, in 3 dimensions, you can see that if you consider the short period of time where you integrate all the images that you see, this XY information is combined with the time information to get the optimal XY coordinate and the optimal time in which you are sensitive to that visual information. So it's kind of confusing, I think, based on some of the questions, how the temporal aspect of the receptive field works. And so just to describe this a little more in detail, when they derived this experimentally, they essentially played a bunch of frequencies over time, and then found out that the response to those frequencies occurred after the point in which the frequencies were initially applied. And one point to note about this is, because you're combining all of the firing rates to certain time points and frequencies linearly using this weight, this assumes that they combine in a linear manner. So if it's a nonlinear function trying to be modeled, it can't be modeled in this way because you're combining all the weights and all the inputs linearly. So to touch on that, just for simple perceptrons in general, from your second midterm, you know that these can't be solved by, these can't solve linear or nonlinear functions. And so an example of this is the XOR, where you have these -1 and -1 outputs for same same inputs, and then 1 outputs for different inputs. And so there's no line that you can draw on this graph to separate the filled-in circles from the non-filled-in circles. And so this can't be solved by a simple perceptron. So the way to actually execute this is to incorporate a multi-layer, a hidden layer in the perceptron to add a unit in order to compensate for the fact that there's not a line that can be drawn here. So this is just one example of one hidden, multi-layer perceptron that solves the XOR problem. You can derive different weights based on which weights that you pick. In your second midterm, you derived what I think this final node should have been. But if you're given any one of these, if one of these is missing, you should be able to derive what the actual number should be based on the output. So just kind of an overview of why this works is that incorporating a hidden layer into the perceptron allows for any function to be approximated because it can be linearly combined with other functions using these basis functions. So one example of this is the sigmoid nonlinear function, which basically is 2 step functions at different points, and then they're subtracted so that you get this this kind of square plot. And then by minimizing the bin width of this, you can model different functions and approximate different functions if you decrease the bin width as much as possible. And that's all for Zhang's review. Any questions? Yes?
Can you go back before that? Can you show how to calculate the output given an input for
Yes. So, for example, if this input and this input are given, you'd multiply the input here by the weight. So multiply this input by 1. And then multiply this input by 1, and then you sum these at the node. So you get this multiplication by this multiplication, and then this number in here, this 1.5, is the threshold, dependent on the model given. So if those numbers after being multiplied are greater than the threshold, then the output is a 1. If not, it is a 0. Well, dependent on the actual function given. But I think for your test it was a 1 and a 0. And then you have a new output, a 1 or a 0 here. And so you multiply that output by -2, the new weight. And then you also add the inputs multiplied by these weights. So at this point you have the hidden layer times the weight, and then also the inputs times the weight. And then if that value, that sum, is greater than 0.5, then the result is 1. And if it's less than 0.5, the result is 0. Does that make sense? Any questions? Okay. Yeah?
I remember on the second exam, when we had to write the equation for that, there was some point where you had to multiply by the step function. Where would that go in applications when you're representing?
I don't think you had to multiply the step function. I think it was the gain applied. Yeah, so the gain is essentially just like the threshold function. So it was like the threshold. That function was defined on the test, but basically it was just what you apply at this node to get the output, and what you apply at this node to get the output. So I think it was like g of x minus h, which was h was the threshold or something. So that would be applied here at the 0.5 output unit, and then here at the 1.5 hidden unit. Does that make sense? And I think those, yeah, I guess they weren't posted. Never mind. Any other questions? Okay, cool.
Should I put it on the drive? Is that okay? Just so you don't have to move around.
On the drive.
Yeah, if you go back. I put it in our folder. So if you go. No, I think it's
Outside.
it's this one. Do I need to hold it?
Oh, or clip it around.
Is this like this one?
It's this way.
Oh, sorry. What am I doing? What do I need to do? Just hold it?
Okay, this one. This is for the recording.
Okay, guys. So for this, my part of the review session, I'm just going to quickly go over Dr. Wong's new stuff. So somatosensory system and auditory system. I'm going to do a little bit of the review of the auditory system as well, which might overlap a bit with Yushi's, but that's not a problem. And before I get into that, though, I want to just clarify something, because one thing that's important to think about for the exam is how different, by basically finding unifying principles across different sensory systems. And I would really be attentive for what similarities are there and how would you compare different features between them. And so I just wanted to go, this is not a slide we strictly went over, but just to clarify some confusion. In terms of basic principles of sensory system, there's these 6 main steps that happens regardless of what type of sensation we're talking about. So the first 2 are the ones that seem to cause a little bit of confusion on exams. So transduction and conduction are 2 different things, right? So transduction is converting energy into a change in membrane potential. So that first step in the perception pathway, and that change in membrane potential is usually not an action potential. It's very rarely an action potential. In fact, when we talk about transduction, we talk about usually an EPSP, right? You guys learned about it in SP1. I was there, so I know you learned it. And usually it's like a small change in membrane potential, small depolarization. And only at the next step, the next cell population, usually a ganglion cell, do you have conduction of information to the CNS with action potential. So I know in the last exam we asked what's the first place along, I forget, it was the visual system where we generate action potentials. It's important to know that it's at the ganglion cell, because that's the conduction is different from transduction. Conduction is different from transduction. And then all other steps are also very similar. So if you keep these principles in mind, it should always make sense. You then have to code the information so the neurons in the CNS can interpret it. You start comparing across neuronal populations. You always go through the thalamus, sort of the relay center of the CNS, and then cortex, and then you can process the information there. So these are sort of the basic principles of sensory systems, and I would keep them in mind in terms of knowing what corresponds to what for different sensory systems we talk about. And so this is the key distinction that for the 2 systems we're going to be focusing on here today. So the interesting thing here is that for the auditory system, it's this very canonical example where you have a hair cell where the transduction takes place, okay? And then you have the spiral ganglion cell, which is the conduction. But the somatosensory is the special case where you have one ganglion cell that does both. That's the exception. And so, and with that very clear dividing line of conduction from the PNS into the CNS. So just make sure you know that at this first step here, for the auditory system, we're not generating an action potential, okay? Because we're only doing transduction. Does that make sense? Okay, cool. So, just getting to the somatosensory system here. In the interest of time, I'm going to kind of go through these slides pretty quickly, and stop me if you have any questions, because some of this stuff is should be pretty intuitive. So first thing I just wanted to clarify is just look again at the somatosensory ganglia. Important thing here is with the CNS-PNS division is you have this the cell body of the somatosensory ganglion cell. You have two branches. One goes into the periphery, so into the sense organ up. Well, I can use the mouse. A sense organ here, where you actually have all your Pacinian corpuscles and all these somatosensory receptors. And then you have one branch that goes into the CNS, which does the conduction to the CNS. Okay, so basically this principle of two branches, which is, if we go back to Dr. Schramm's first lecture of the entire course, this is the sort of principle he was talking about. Okay, where do you actually find these ganglion cells? So the cell bodies are in these pairs of dorsal root ganglia along the spinal cord. Just that's location-wise where they are. And then if you go back to the periphery here, what we're talking about, the somatosensory receptor part, there's these different types at different depths in the skin that we focused on. So the four ones we talked about are highlighted here with the arrows. So we have Meissner, Merkel, Ruffini, and Pacinian. Ruffini we don't really talk about, and it's very poorly understood. But the idea here is that these different types of receptors respond to different types of energy, or different types of environmental energies. They have different sort of channels that determine the sort of response, even though we didn't really get into that much. And these are the basis. The fact that we have these different receptors are the basis for the different types, different aspects of somatosensory perception that we have. So that's a very big overview of the somatosensory receptors. But when we talk about the afferents, so in other words, the sort of like what the receptors are connected to, essentially. Here's a diagram just clarifying this division between rapidly adapting and slowly adapting. I just wanted to make sure it's clear where that comes from. And so when we talk about rapidly adapting and slowly adapting, what we're talking about is if you have a blunt probe and you indent the skin by like a centimeter or 2 centimeters or so, the mechanoreceptors you have in the skin that we talked about respond, or fire action potentials in response to the mechanical energy in different ways. And so when we talk about rapidly adapting and slowly adapting, it's literally how they adapt to the presence of that stimulus. So if you look at the rapidly adapting here, here's the stimulus. And as you maintain that, it initially fires when you apply the stimulus, but it stops firing right away. So it adapts immediately to the stimulus. Slowly adapting, it keeps firing. So that's the basic distinction. And then within rapidly adapting and slow adapting, you have like subcategories. Okay, so within rapidly adapting, you have this RA, which means basically they're driven by rapid application and removal of the stimulus. And the only difference for the Pacinian is that it's still a type of rapidly adapting, but the application of the stimulus is more, there's a higher frequency, so it's basically a vibration. But it still falls under the category of rapidly adapting. And then slowly adapting, again, all this means is how does it generate action potentials in response to this probe being indented into the skin. And it takes a while to adapt. And there's two types, SA1 and SA2. And as we'll see, those match up with Meissner and Ruffini. And so the key difference there, even though we didn't really talk about it, has to do with their levels of spontaneous activity and just how slowly they adapt. So SA2 are basically super slow adapting and very low spontaneous activity. And SA1, basically no spontaneous activity, but adapt slowly but less slowly than SA2. But does this distinction kind of make sense? Rapidly adapting, slowly adapting, pretty intuitive. And then the types of receptors and the afferents they connect to fall into those subcategories. Okay, cool. So just a nice diagram showing some distinctions between somatosensory afferents and how we can classify them. It's good to know. Obviously the axon diameter is tied to the conduction velocity, and as you see here, the muscle spindle isn't really what we're talking about here, but our sort of somatosensory perception that we focus on for this section is this touch row. And so just relative, it's good to know relative to the other sorts of stimuli the sensory afferents are able to picking up, how it compares. So it's a good contextualization slide. And then here, what we're trying to begin to tie in is the types of receptors and their receptive field size. Again, at the bottom here, you see evidence of the rapidly adapting and slowly adapting thing, right? You have the stimulus that's the same every time. Rapidly adapting, it stops firing as soon as the stimulus is continued, and slowly adapting, it takes a while. And so here you can see what I was saying about rapidly adapting and slowly adapting subcategories. So all these are representatives of the 4 subcategories, right? You have SA1, SA2, PC, which is just Pacinian corpuscle, and the RA, the rapidly adapting RA. And so what it's trying to show here is, this is just a reminder that you can kind of capture these differences between rapidly adapting afferents using a basically like the psychophysics setup we talked about in the beginning of the first part of the course. And so what you see here is what I was saying, right? The difference between RA and PC is that the vibratory frequency at which you get the, the the minimum amplitude you need to drive it it happens at a higher vibratory frequency for the Pacinian. Yep.
The receptive field is related to the density of receptors, right?
Yeah, yeah, that's exactly, the innervation density, and I'll have a slide showing that nicely. Yeah. Any other questions? It's pretty intuitive, and so this is exactly what you're asking about. So innervation density and receptive field size are obviously very linked. This diagram here just shows that what it's trying to quantify is the density of Meissner and Merkel cells, okay, with the blue, and the green is the spatial resolution, and basically, or the acuity essentially, and basically they're tied together. As you go from the tip of your finger upwards up your arm, you get less receptor density, which means what ends up happening is increased receptive field size and decreased spatial resolution. Yep.
Does the intensity of the stimulation threshold in order to sense it change with receptor density? The threshold. For like an individual receptor? Yeah.
I don't believe so. For an individual one, no. Yeah, I would think about it in terms of populations, but the idea, I don't think threshold would be effective. Yeah. Any questions? Okay, so this is a key idea, and then this is a slide from Dr. Wang's slides that kind of just shows that, and in general, rapidly adapting and slowly adapting, there's a very clear distinction there between the receptive field size. And this slide, if you want to like take one slide from this first part, I would just note this slide. It captures a lot of the previous slides. This is not in the notes, but it's from a class I took. And basically it captures everything, right? The rapidly adapting, slowly adapting, the receptive field size, how it compares. And these 4 are the canonical examples we use for each of the subcategories, where they're positioned depth-wise, the type of stimulus they respond to, all this stuff. So basically, if you know these 4 columns, you should be in pretty good shape for this part of the course. Make sense? So, yeah. So then when we talk about going on to the cortex part of it, so now we talked about afferents, what happens at the cortex level. The somatotopic organization of S1 is something Dr. Wang briefly touches on. It's found in this part of this region of the brain, which is let me just bring up the anatomical. It's it's a post-central gyrus, for those of you who are interested. And the idea is that something we've talked about before, that basically the areas which are finally controlled have larger portions of the somatosensory cortex dedicated to them. And this is where this homunculus comes from. Basically, if the actual human body, like the ratios of different body parts matched up with the amount of cortex in S1 dedicated to them, this is what a human would look like. Something you've seen before. And the final part of somatosensory here for the review session is this idea of plasticity. Dr. Wang didn't discuss this extensively this year, but the idea is that what it's defined as long-lasting modifications in neuronal response that accompany changes in stimuli. Usually you see it either as a result of learning, or in this case as a result of an injury. So here's an amputation example. where basically you get these topographic changes in sensory representation. So if you get rid of the third digit is amputated, basically the S1, the somatosensory cortex, there's other areas basically take over that area of cortex, and that's what we mean by plasticity, and those changes are long lasting. And so he doesn't really discuss anything much more mechanistic than that, but definitely understand that key idea, the overarching idea, and know what can lead to plasticity. And so we obviously a good question might be linking this to some of Dr. Kirkwood's stuff, because there's a lot of elements of plasticity in learning as well. That's somatosensory wise what changes are happening in S1. That's just a brief overview. So I think that's it for somatosensory. So any questions on somatosensory? This was just one lecture, so it's relatively short. Cool. So moving on to auditory. So this is quite a bit more stuff. I'm going to go through the more basics pretty quickly, because I think Yishi might touch on them as well, and I don't want to be repetitive. But definitely know the basic anatomy of the ear and the auditory pathway in general. I'm not going to go through all of the anatomy here, but I would definitely know especially well the anatomy of the cochlea, specifically the arrangement of the frequencies along the basilar membrane and the arrangement of inner and outer hair cells. Definitely know this idea of the afferent and efferent information carried by either of them, or by both of them. And one topic that hasn't really come up, but that's important to know, and I think Yishi might cover this, is just the role outer hair cells play in modulating adaptation to your sound environment, and they act back on the other type of hair cells. So the basic anatomy, definitely responsible for it, especially the cochlea. Just a quick overview of how exactly this works. So you have the sound wave, it's collected at the pinna, vibration takes place at the eardrum, you amplify it with the three little bones. And then in the cochlea, where we usually focus our attention, you have fluid there, endolymph, perilymph, and one more, I forget which one. The fluid inside vibrates, and then you basically get this vibration up and down of the basilar membrane, which definitely know this is something you should be very familiar with. This frequency map essentially along the basilar membrane. So you have this narrow base, which is tuned for higher frequencies, and as you go towards the apex, you go to lower frequencies. And then also definitely know the basic mechanism of how we're first doing the transduction. So if you remember the transduction, so remember the mechanically gated ion channels at the tips of the stereocilia of the hair cell, and caused by shear force. One way, if they tilt one way, they open the channel. Tilt the other way, they close the channels. It's potassium influx. You get a depolarization, but not an action potential. If you guys get that wrong, I'll be really sad. And then you release neurotransmitter into the basically the ganglion cell, which we'll talk about now. Okay, so this is the basic overview here. And again, action potential generated at the auditory nerve ending or the ganglion cell. So again, don't forget this. You have hair cells responsible for transduction. So basically all we're doing is converting the sound energy, so the basic information of the sound wave, into a change in memory potential, which then causes release of neurotransmitter into the spiral ganglion cells. And those are the ones who actually transmit the information or conduct the information to the CNS. Okay, so make sure you don't forget that. Okay, so now we can start talking about speech representation. So in the auditory nerve, there's two basic ideas to think about. One is this idea that you can represent speech, or the basic elements of speech in the auditory nerve, by firing rate. So there's a low population of neurons that are we're talking really about ganglion cells that are low spontaneous, and a population that are high spontaneous. Again, pretty intuitive. It's exactly what the terms imply. So depending on the firing rate of those two types of populations, you're able to encode some information. And the other main way is through this mechanism of phase locking. And as we'll see, it's something that, although it degrades, it's maintained at higher levels. And this is just a reminder here. I won't dwell too much on this because we've talked about it before, but just definitely a reminder that this is definitely a part of the course where Dr. Wang can tie it back to some of the earlier stuff in terms of period histograms and all that. And you should be very familiar with how to tell if something is phase locked by looking at the period histogram. Yep.
Higher levels, you mean intensity or frequency?
Maintained at higher levels of, no, sorry, but higher levels of like processing, like along the auditory pathway. Yeah. And also intensity of sound as well, depending on where you are. Okay, so I just wanted to, this is a slide that again we didn't strictly cover, but I added it because I think it's important to know, to like understand what's going on. So just to understand like the biological basis of phase locking. So where does like what do we talk about when we talk about phase locking? So the idea is so along the basilar membrane, right, in the cochlea, we have this tonotopic map, you know, from high frequency to low frequency, and different populations of hair cells are along that basilar membrane. And those populations of hair cells are tuned, because of where they are, they're tuned to a specific frequency. And those populations of hair cells communicate with a specific population of ganglion cells, okay? And so the idea here in this tuning curve is that depending on the depending on where the ganglion cell is, and more specifically the hair cells it innervates, different ganglion cells are tuned to different frequencies, okay? And this is a place code that's maintained into a few more stages, right? So if you go at the basilar membrane stage, right, hair cells at a particular location tied to a particular frequency communicate with a particular group of ganglion cells, and that map is maintained in the cochlear nuclei. So basically, depending on where you are at each of these steps, you know what range of frequencies that corresponds to, because it is tied to a specific place on the basilar membrane, which is tied to a specific type of hair cell, which is tied to a specific type of ganglion cell, and so on. So it's a place code. And so where phase locking comes in is below a certain threshold frequencies, not only do ganglion cells fire in response to the sound wave that we talked about, but they fire at a particular part of the wave, okay? And that's what it means by phase lock. So if you're under a particular frequency, a particular part of the sound wave, I fire every time at the same point. And the idea here is that allows you to have much more precise information, because ultimately when you're trying to distinguish between, let's say, a sound of 300 hertz and 301 hertz, you know that that's a relatively low frequency, so you can use place code information as well as temporal code information, because the firing of the ganglion cell will be locked to the frequency of the sound wave. But above a certain threshold frequency, you can no longer phase lock, and so that's why it's much easier to distinguish between, or you can distinguish between 300 hertz and 301 hertz, but you can't distinguish between 3000 and 3001 hertz, because at those levels of because that difference between the ganglion cell populations is very, very small, right? When we're talking about 1 hertz difference, the population of hair cells that you're stimulating at the basilar membrane is basically the same. It's the same region. But if you additionally have it phase locked, then you can do something as simple as, if you have, you can match up basically the number of peaks of the firing of the ganglion cell with the number of peaks in the sound wave, for example. And you're able to tell, okay, 300 versus 301, 301 peaks versus 300, like big pictures, just so you guys understand. But 3000 hertz versus 3001 hertz, you can no longer, wait, don't. Did it die? Okay, a little setback. But basically I can, 3000 hertz versus 3001 hertz, the idea is, I don't have HDMI though. Do you have HDMI?
Take a few minutes.
I think Andy, does Andy have HDMI? Where do you go? But my computer, I don't trust it. Yeah, let's just get his laptop. Okay, but I'll just there'll be a little delay here, but I'll just explain that last point. And so, does that make sense to you guys? So if I have 3000 versus 3001 hertz, and I can't phase lock, so the only way I can distinguish between those two frequencies is by using the place code, but that's not really enough because the population of hair cells that I'm stimulating and the population of ganglion cells that I'm stimulating is basically the same. Okay, that's the idea. That's why you want phase locking. And can anyone? I briefly told these people in my section this, can anyone kind of infer like what the biological basis for the frequency cutoff is, like biologically? If you think about the hair cell ganglion cell connection, why is it that at a particular frequency you can no longer phase lock in terms of the firing of the ganglion cell? Not really refractory period, no. It's the, well, what's another guess? So what is causing the ganglion cell to fire? The release of neurotransmitter from the hair cell, right? And what's causing that release of neurotransmitter? The opening and closing of the channels, right? And so if above a certain frequency, there's no longer, it's not really opening and closing, it's just open all the time. And so you can't really have, so the reason it fires, the reason it phase locks is because it's tied to the release of neurotransmitter. But if the release is constant because the tips of the gates are always open, then you won't have that firing, that locked firing. But I don't think you'll ask that, but it's interesting to know. Okay. Okay. Okay, so moving on. So if we go, so that was auditory nerve stuff. If we talk about auditory cortex, there's two main points in this slide. One is definitely know this basic pathway. So basically, cochlea, auditory nerve, cochlear nucleus. Cochlea, auditory nerve, MSO-LSO. In other words, the superior olive, cochlear nucleus, and then inferior colliculus, thalamus, cortex. That I would know because he explicitly does compare the stimulus selectivity across these different levels. But the basic idea here, without dwelling too much on the experiments, because I don't want to waste too much time, is auditory cortex neurons are usually not driven by simple stimuli. You need a complex stimulus to drive it. And so there's a series of experiments that Dr. Wang describes where basically different combinations of tones with different frequencies drive a particular population of auditory cortex neurons. And we can put the reference for you guys to check out those experiments, but that's the basic idea. And this is a recurring motif, essentially theme throughout the auditory pathway. So essentially, if you start out at the auditory nerve and move upwards towards the auditory cortex, the stimulus selectivity progressively increases. So the type of stimulus you need to drive the cells in that part are increasingly more complex. Yep.
So the blue part, what's onset firing? Are they just not firing, or there's a non-preferred stimuli onset firing? It's a little. The term is a little confusing, but basically it's what it says here. So like a preferred stimulus causes like sustained firing in the sense that like, so onset firing, yeah, basically like I would think about it as like the background, firing, and then basically background firing versus the firing you would get from a response to that particular type of stimulus. And the idea that it's making is that as you go up, the stimulus needs to be more and more specific. Yep.
You went into depth about some of the stuff that they discovered, I think very recently, about the two-bit stimulus for the cortex, auditory cortex. Did that matter too much, do you think, or was it just an example saying it's very difficult to stimulate the neurons in the cortex?
I mean, I can't speak for Dr. Wang, but if I were a student, no, I am a student, but if I were a student taking this class, I would go back and re-listen to what he said, but I wouldn't worry too much about it because I don't think he's going to ask like explain the 2-pip experiment in fine detail. But it might come up like I don't know, like this idea of stimulus selectivity. And also like these review session slides are big picture ideas. But in answer to your question, I would re-listen to it to like better understand it, but I wouldn't worry too much about it. And so this is the basic idea then, this is a recurring theme. This makes sense? So basically stimulus selectivity increases as you go up. And so now going back to this idea of phase locking. So this is a really important slide. One is this idea of phase lock. So when we're trying to represent the temporal information in speech, one idea is this idea of phase locking degradation. So if we talk about the auditory nerve, based on this curve here, at about, I'm going to say like 3 or 4 or whatever this is, kilohertz, you no longer, so sorry, before I say that, you have here on the x-axis you have frequency, and on the y-axis you have this vector strength, which is just a quantitative measure of basically phase locking. And so the idea is that this shows that as you go up so if you go from right to left, you're going up the auditory pathway, you get phase locking degradation, meaning the threshold frequency until which you can phase lock decreases as you go down. So basically, for example, auditory cortex, the last stage we talk about, you can no longer phase lock after about like, it seems like a couple hundred hertz or so, or something like that. While at the auditory nerve, you can do it into a few thousand hertz. And another key idea here is that speech information, you have speech information has very complex sound information, and depending on the so you basically you extract different features out of speech based on the frequency, based on like the types of frequencies that make it up, and the basic classification is this one he has here, where below 50 hertz for like recognition of syllables and words, you have this envelope, and then periodicity, and then fine structure. The idea being different timescales of sound are you can categorize them in this way based on the frequency information. Okay, let me just make sure I'm not forgetting anything for that. Yeah, so speech contains information distributed across multiple frequencies that span in time scales from few to tens or hundreds of milliseconds, and you can categorize it in that way and extract information that way. And so, does this kind of make sense? Sorry. Yeah.
Yeah, and one of your first slide about phase locking, you mentioned that it was preserved at higher processing centers, but this seems like kind of
it's preserved like the incident, like phase locking happens, but it degrades. That's what. Yeah, it's a little confusing. Does that mean it's not preserved in the sense of like it's not phase locked exactly the same way. And also he briefly touches on in his lecture about why this is. So I would think about, sorry, yeah.
I was just going to say, the auditory cortex neurons are a lot more complicated because one neuron is encoding the information from many auditory nerves. So if you have 10 auditory nerves that are phase locked, it's going into one auditory cortex neuron. And the auditory cortex neuron is also phase locked, and it kind of has preserved the phase lock. It's just that.
Yeah. So yeah, exactly. So you can, it's not when I say preserve phase locking, I mean like the phenomenon of phase locking, not necessarily the exact frequency cutoff. But as we'll see in auditory cortex, even though the phase locking limit is so low, you're still able to get out information using a different mechanism, which we'll touch on in the next slide. But yeah, basic idea here is these two. Phase locking degradation especially is very important to make sure you understand. Yeah?
So why does phase locking degradation happen in the auditory cortex?
Oh yeah. So basically what the explanation Dr. Wang gives is that as you go up the auditory pathway, it's more and more, every time you pass on to the next level, it's a synapse. And so what he says is that at each synapse acts as a low pass filter, high pass filter, where basically which explains how each time you go through a synapse, you chop off the highest frequencies you can detect, and the reason why it's a bandpass filter is something. Intuitively sort of makes sense, but I guess like moral biology of that is something I have to look into because I'm not fully sure. But if he asks, I would do this idea of filtering out the frequencies each time you go through a synapse. Okay, any other questions on this? Okay. Okay, so go back to your question. So how do we actually represent temporal formation in the auditory cortex? So there's basically two, populations of auditory cortex neurons that we're interested in. And this goes back to the idea of the frequency of the stimulus we're talking about. So basically, the faster basically as you increase the frequency or basically as you decrease the ICI, the interclick interval, you increase the frequency, and so at a certain point, you're going to go past the threshold frequency for there to be phase locking. So when we talk about synchronized responses, we're talking about neurons in the auditory cortex that phase lock, okay, from the very beginning. And so those are tied to, this is what we mean by a timing code. So if the signal is slow enough, you're able to extract information using phase locking mechanisms. But you see, like after a particular one, so this is interclick interval. So after a particular, once you go to a particular level, eventually you reach here the high frequency cutoff, and so you can no longer use these to extract information from sound. But what you do have in the auditory cortex is this non-synchronized population as well, where there it's just a firing rate based code to represent the rapidly varying signals. And so because you have these two populations, you can encode a wide range of frequencies or interclick intervals, because basically when one dies down, the other kicks in. And so just important to realize here is that the y-axis for these two are different. Okay, the y-axis for when we're talking about the time code is vector strength, so in other words, trying to quantify phase locking. And for the non-synchronized, we're talking about firing rate, okay? And same thing here. This is what I meant by what I said earlier. So basically, if my interclick interval, if I go from right to left, because it's more intuitive, if I go from a really low frequency and progressively increase it, my synchronized population, my ability to phase lock degrades, right, like we talked about, eventually until I reach a point where I can no longer phase lock, and that population is not particularly useful. And the non-synchronized population, because it's dependent on it's coded by firing rate, it's able to, it's maximally, it fires maximally basically at these super high frequencies. Yeah.
And what's it by implicit?
Oh yeah, that's the whole like can of worms. The implicit versus explicit is a little bit confusing because the way Dr. Wang uses implicit and explicit is a little bit different from how we've seen, for example, Dr. Connor use it. But how I think about it, and you guys correct me if I'm not making sense, is that temporal coding, basically you're phase locked to the wave, so you're explicitly coding the sound as it is, like the sound wave as it is. While the firing rate, it's implicit in the sense that it's implicit in the sense that you're not directly tying it back to the exact characteristics of the sound wave. That's more or less how I think about it. But honestly, you don't need the terms implicit and explicit to understand this. Just understand the difference between temporal code and firing rate code, and you should be fine. Does this make sense, this slide, how we get information? Okay, cool. So, moving on. Sound localization, there's basically the idea, just reminding anatomically where we are. We have hair cell, we have the ganglion cell, and now we're at the superior olive, okay? And we have basically a few populations of a few structures to keep track of in the superior olive. So specifically LSO and MSO are how we compute interaural level differences and interaural timing differences, which is exactly what it sounds like, computing differences in time of arrival of a sound wave versus the intensity level of a sound, comparing the two ears. So these are both binaural cues, so use information from both ears to compare. And then he doesn't discuss this explicitly, so I'm not going to dwell too much on this for this review session, but basically the pinna in the outer ear allows you to filter out basically the way you get elevation information out of from sound is using these spectral notches, which basically are different frequencies that you're filtering out depending on the angle where you're coming in, and which frequencies are filtered out by the outer ear, or the pinna. And so we explain this a little bit more in detail in section. I'm not going to dwell too much on that, but the idea is this is a monaural cue, and these are two binaural cues. And this is for level and timing differences, and this is for elevation information. Okay? Okay, so those are the mechanism of ILD. It's very intuitive. All you're doing is comparing ipsilateral with contralateral input, and basically the input you receive ipsilaterally is excitatory, and the input you receive contralaterally is inhibitory. This is shown in this diagram here. So at this point, it's also important to know that superior olive is the first instance where there's kind of this crossing over effect, where you're starting to compare the signals from both ears. And it's shown here by the type of neurotransmitter you receive from the ipsi versus contra ear. One is like a excitatory neurotransmitter, and the other one is inhibitory. And this figure sort of recapitulates that in terms of the response rate as well. So basically, excitatory from ipsilateral, contralateral from, inhibitory from contralateral, and based on the stimuli you're receiving, you can compare the two and compute the interoral level difference. And so ITD, just very quickly, there's two basic models that we discussed in lecture. So the first is this idea of delay lines. This is a Jeffers model from the 1940s. This doesn't apply to mammals, although it's been kind of found to be roughly true in birds. But again, it's relatively it's a clever setup. So basically, depending on where your sound source is, it'll reach one ear before the other. And let's say it reaches the left ear first. Here's the cochlea, obviously, and then we're generating an action potential that begins to travel towards the MSO. And not simultaneously, but a little bit shortly after, the sound will reach the other ear, and again you'll begin to fire action potentials. And Jeffers' model is that, so these are what we call delay lines, okay? Basically the pathway from the cochlea and the ganglion cell into the MSO in this case. And what we have here are coincidence detectors. So delay lines and coincidence detectors at the MSO. And his whole model is that depending on where the MSO neurons are located, they're essentially tuned to a particular timing difference. So whatever the delay is whatever the difference in time of arrival of the information from the ganglion cell is between here and here, that population of neurons is tuned to that difference. So there's a map of ITD. So not only do neurons respond to characteristic delays. So if I go into the MSO and probe a neuron, not only do they respond to characteristic delays, but there's a map. There's a map of in terms of how they're positioned, lining up with the different delays. And so basically in mammals, what we found is that this is not quite the case. In mammals, anatomically what's going on is that a single MSO neuron receives receives bilateral excitatory inputs and bilateral inhibitory inputs, and by some mechanism that's still not super clear, the interaction between those four inputs generates the ITD sensitivity. But the idea, so the key idea to understand is why delay lines don't apply to mammals is that, so if you go into a mammal's MSO and you stimulate the neurons, you do see characteristic delays. In other words, you do see neurons tuned to particular delays, but they're not arranged in this map that we talked about with the the previous model. Does that make sense? So there's still characteristic delays, but they're not arranged in a logical map. So it's not like they're arranged in this very organized way, but they do respond to characteristic delays. So there's some comparison of timing differences going on, and basically this model that Dr. Wang briefly presents is this two-state model, where I'm not going to get into it like too much now, but the idea is that perhaps one possibility is you compare the MSO signal, so to speak, from both ears, and based on the difference, you're able to establish whether you're able to compute ITD Let me just make sure that I'm not missing any key detail. So basically the idea is that sound source at the midline drives equal numbers of neurons to be equally active in the two MSOs, but as the sound source moves to one side or the other, the total activity in the contralateral MSO increases, while that of the ipsilateral MSO decreases. And so you sort of get a code for the location of a sound source. So a little speculative, it's nothing set in stone, but it's one possibility. And one question I'd definitely be prepared to answer is comparing the ley line or Jeffers model with the two-state model.
What's comparing the MSOs again? There's another sector that's comparing the population.
How it's compared is not very specified, but the idea is by some unknown mechanism you compare the population of neurons in both MSOs to try to extract localization information.
Previously it said after the MSO the information goes to the cochlear nucleus, but it's not done there. And after that would be pretty crucial.
Cochlear nucleus is before MSO.
Oh.
Yeah.
So the higher space should be high C.
The inferior colliculus. Yeah, I might have switched the order by accident. Okay, does that sort of make sense? So definitely be familiar with those two models. Okay, so last part of the Dr. Wang section, just some relating to pitch and harmonics. So for pitch, the first key thing is just some basic definitions. Here you have if you just look at speech information, you can represent in terms of the spectra, or in terms of and like temporally or in terms of the spectra. So like you can just do the Fourier transform. And the idea is that the first peak you plot here is the leftmost peak here is just 1 over the period of the temporal profile, which is defined as the pitch. So this is like 1 over the pitch. This is the period here of the temporal profile is the pitch, and each other component here in this profile is just a multiple of the pitch of the value the multiple of that fundamental. So we call this the first fundamental, which 1 over the pitch, and then each of the gaps between them is equivalent. It's the same value of the pitch. The interval between harmonics we call the interval between harmonics is the pitch, and the 3 peaks you see here are the formants, and this is because this is the information for a particular vowel sound shown here. I think it's or something like that. Basically the key thing to understand about formants is that the different peaks you get, the different formants you get, are characteristic of a particular vowel sound. Yeah.
So the pitch temporal would be in the envelope, right, of the sound wave?
I mean that depends on what frequency that would be. Yeah, but
right.
Yeah. But I mean, more basic than that. Just like generically, what the pitch is the period of the temporal profile, and biologically, where you determine pitch is by the vibration frequency of this vocal fold here. Okay, and like you're saying, the envelope and the formants, you determine that by depending on the shape of the vocal tract. And so he explained this in a little bit more detail in the lecture, but I'm not going to get into it now. Key ideas here, and this I'm going to breeze through because I want to leave time for Yushi. First thing is, in the terms of the harmonics, those are captured pretty faithfully by the auditory nerve firing. But at the auditory nerve, we're not extracting pitch information yet. Okay, but you are able to capture the harmonic information, or at least represent the spectral harmonics. Yeah, and then where we actually do represent pitch is in the auditory cortex. And so what these diagrams show is so basically, pitch information is extracted in the auditory cortex. And the key features to understand here is that if you look at a pitch versus a non-pitch neuron, the non-pitch neuron only responds to a particular a sound with frequency components that fall into its receptive field. So for example, usually pure tones is what we're talking about. And a pitch neuron fires in response to, but it doesn't fire in response to harmonic complex stimuli, meaning when you start getting multiples of that, but that are outside the receptive field that that particular neuron is tuned to. But pitch neurons fire both in response to the fundamental that it matches up with the pitch, as well as the multiples of that, the harmonics of that are technically outside its receptive field. It still fires. So they're also tuned to pure tones, but they respond to harmonics complex tones when all spectral components are out of its receptive field, but the space between harmonics matches up with the pitch. And this is a similar. The graph shows a similar sort of idea. And so does that make sense? Pitch neurons. Okay. And so harmonic neurons are a little bit complicated, but the idea is that elsewhere in the cortex, you have neurons that are sensitive to the harmonic structure of the sound spectrum, and you get a greater response when you play harmonic complex tones. And so the idea here is that, for example, if you have, and you should correct me if I say this wrong, but basically at particular parts of the auditory cortex, if you have let's say you have a particular characteristic frequency here of 7.49 kilohertz, you get an enhanced response at the harmonic neurons or the neuron fires most when, for example, the 7th harmonic of this 1.05 when you observe the 7th harmonic of that 1.05 or the 6th harmonic of the 1.26. So basically, it is able to capture. It fires when if the fundamental frequency, a particular harmonic of the fundamental frequency lines up with its characteristic frequency. It's able to pick up that sort of information. And again, details of this experiment, similar before, I'd go back and rewatch Wang's lecture for additional detail, but the key idea is here. And one thing I would know for sure that's definitely a possible question is comparing the two. What are some differences between the two? Again, pitch neuron, they respond even if all the frequency components are out of its receptive field because it's tied to the pitch. But the harmonic neurons do require the frequency components to be in its receptive field, so the 7.49 or whatever the case was for the example. But you do get an enhanced response when the spectrum is harmonically related. And the other thing is that the pitch neurons are found in a very small restricted region of the brain, essentially around the auditory cortex. But harmonic template neurons are found all over, and the speculation is that, and this is like the model that's shown here, is that it receives long-range horizontal inputs from other locations with these CF2 to CF regions. I wouldn't worry too much about that. Difference being that you have many more, or like they're much more spread out across the auditory cortex as compared to pitch neurons. And so, does that make sense, these differences? Yeah.
For a single harmonic neuron, can there be different fundamental tones played with the harmonic spectrum it responds to?
Wait, say that again? So if you have a harmonic neuron, yeah?
Particular harmonic spectrum with different fundamental period, or does it have to be a specific fundamental period and then like the 6th and 7th harmonics on it?
I think my understanding, and correct me if I'm wrong, sorry?
It could be different.
Yeah.
So as long as it has the 6th and 7th harmonics, for example, it could have any fundamental.
As long as it lines up, my understanding is that as long as the harmonic pattern at some point lines up with its characteristic frequency, it'll cause enhanced firing. Does that make sense? Yeah, sorry, yeah. Yeah, exactly, yeah. But as long as it, yeah, exactly. But it has to be in the receptive field. That's the point of the first bullet. And so this is just, and the final part of my review is, damn, I'm going really long, sorry, one thing is just to keep track of all the different areas of the brain we're talking about. So for speech production, we're talking about Broca's area, speech comprehension, Wernicke's area, and then we have somatosensory cortex and right near motor cortex, and a pitch center is just this tiny dot over here. So just this final part of the review. The key idea is we're starting to talk about vocal control and basically what happens when in the auditory cortex when you speak, okay, and when you speak and how you make sense of your own voice. The first basic idea is that in general, the auditory cortex is suppressed when you speak, and specifically what you find is that before I get into that, the idea is that when we speak, the sound of our voice is delivered to a listener, but then it's conducted back into our own ear, and the feedback, this is what we mean by feedback, we'll keep using the word feedback, is a major input to our auditory system during vocal production, and is constantly self-monitored, which requires the detection of the feedback changes by particular neurons in the auditory cortex. And so, if you go into auditory cortex and you record from it, when you're speaking, and this is what I mean by onset and offset of vocal onset, you see that most auditory neuron cortex, most neurons in auditory cortex is about 75%. You get inhibition or suppression of neural firing during vocalization, during when you speak. There's a smaller subpopulation as well, here, about 25%, that actually are excited during self-initiated vocalizations. And so that's what's shown here. You have the same onset and offset of a particular vocalization, and in this population of neurons in the auditory cortex, it stops firing altogether, and this other population, it fires massively. But the majority is this one. So you definitely have two different populations, and what's interesting here is that this suppression effect is very different from the typical response you get when you play back like a particular sound, because if you play it externally, then the response is always excitatory. If it falls into neurons, it falls into the receptive field of neurons that are tied to the frequency information of that stimulus. So basically this inhibition effect you see here only is like very different from what you would see if the sound was played externally. Okay, but there's definitely two populations, with this one being the majority. Okay, makes sense. And so then what they did here, I'm going to skip the experimental stuff because in the interest of time, is that basically if you run these experiments where you start shifting the frequencies of the feedback, you observe some really interesting trends. So you're looking at here you're looking at the suppressed neuron population, meaning this first population here, and then the second graph you're looking at the excited neuron population, and you find that if you change the frequency of if you shift the frequency of the feedback, you can start affecting the response. So the population here, if you shift the feedback, the suppressed neurons actually fire more, but the excitatory neurons aren't really affected by the shifted feedback. And what you see here is that one example of a neuron is you get this massive increase in firing rate with the frequency shifted, but if you have it amplified or baseline, there's no response at all. And so the idea here is you alter the perceived feedback the animal has. This is what the funny picture with the marmoset with headphones on. You shift the frequency that the animal perceives as feedback, and you see that depending on the population of neurons you have, the response can change. And so how we account for that is with this is my last slide, with this forward model setup, where we basically have this control loop, and we have basically, depending on the two scenarios we have, or external sound is being played or a self-produced vocalization is taking place. And the idea is that if in the normal case is where there's really no comparison between, there's no prediction between the expected auditory input as compared to the actual feedback you would expect. But when you have a self-produced sound, you have this mechanism taking place where these efferents copies or modulatory signals contain predictions of the expected auditory signal, and you compare that to the actual vocal feedback you get from the sound coming back to your ear when you speak. And the auditory cortex activity simply reflects the difference between the efferents copy and the feedback. And if that error is greater than 0, then you get the vocal control center will change its command and tend to produce a different pitch to compensate for the error. So the change of pitch will be in the opposite direction to the direction of feedback shift. And the forward model, basically what it does is it modulates change. So this is when you have like suppressed neurons in auditory cortex, when you have this change here, okay, basically becoming less suppressed, that's basically captured here. So the forward model, the suppressed neurons in the auditory cortex are less suppressed if the error signal is different, because you're changing the feedback. In the experiment, basically you're changing this feedback component, which will lead to a different error signal, which when it feeds back in here will modulate it differently and so possibly suppress less. Okay, that's the basic idea of how we try to compare the two signals and control our pitch accordingly. Any questions on this? Okay, cool. Sorry it took so long. Sorry, machine. Just go over like the lists. I don't think you see
where is it? I guess we have 40 minutes left because this room is reserved until 1 p.m. Okay, so I'll first go through of my own lecture. So maybe this part will only take about less than 10 points, and I haven't think about what question to give. But I basically introduced another technique to record neural activity. So previously we have been talking about electrophysiology, which is extracellular recording or intracellular recording, and we also talk about fMRI, and this is another way to image neural activity. So I think the key point is to understand how do we image neural activity. So first of all, we have to label those neurons to make the neurons give out some light. Then we can use a microscope to see what is going on. Okay, then we introduce two types of microscope. One is confocal, one is two-photon. Okay, let's first talk about the label part. So the stuff we used to label the neuron is called fluorescence, and there are some basic physical principles for how does a fluorescence work. So basically for fluorescence molecule, you have two processes. Yeah.
I just had a question. I noticed the lecture you posted, there's a slide where it says you didn't get past this point in the lecture. Is the material before that on the exam, or is the material after?
Before.
Just
like. So there are two processes for fluorescence to work. One is you have to give some light to excite, and to excite the molecule to a upper excited state. Then after that, the neuron or the molecule will give out another light, which is the emission process. So the thing to notice is you excite with a higher energy light, which is a shorter wavelength. So in this case, it's blue. And when it's emitting another light, then it becomes a longer wavelength. So it gives out a smaller energy light. So this is the two process. And this shows you how does a two-photon absorption work. So in this case, you can excite this molecule with two much longer wavelengths. So if the molecule can observe those two photons together, then it can still be excited. Then go to this process, emit green fluorescence. Yeah.
So it's kind of similar to like a coincidence?
Yeah, something like that. But you need to be very coincident. Okay, so then the idea is you can put these molecules in a neuron, and the way people do it is usually through some viral transfection or transgenic techniques, and you can select which group of neurons you want to label. So in this case, it shows you a commonly used fluorescent or calcium sensor. So you combine something with this called GFP, which is green fluorescence protein, and combine this GFP with some other, this blue part is a calcium sensor. So when there's no calcium or only a few calcium in the cell, then this GFP is not activated. So this fluorescence doesn't work. Once you have some action potential, then some like voltage gated calcium channel will open, then you have suddenly a huge increase of calcium concentration inside of the cell, then this molecule will be activated. So in this case, if you shine some blue light on this molecule, you can see green light emitting from that cell. Okay, so this just show you some dynamics of this GFP, or in this case, you can only focus on the black curve, which is the most recently used called GCamp6S. So this is when you have an action potential, for example here, then you can see this y-axis, the fluorescence change. So this is delta F over F0. So F0 is just the baseline. And you can see that if you have one action potential, then it corresponds to about 30% of fluorescence change. And if you have 10 action potentials, then this change could be 500%. Okay, so that's what else? So the whole process is you have action potential, then calcium influx, then activate those molecules, and you can see the GFP working. Okay, then this is the general structure of a microscope. And the key thing to understand is any microscope have a resolution limit, and this limit comes from some diffraction phenomenon. So if you visualize this is called PSF, point spread function. It looks like a 3D structure, and this width of this PSF depends on some wavelengths and the numerical aperture of the objective you're using. So you don't have to remember all those equations, just get an idea that you're not focusing on a infinitesimally small point under the microscope. And this is a figure showing you if you have multiple dots in your sample, then what you actually see is a convolution of those real image and this PSF function. So you have some certain limit of how close you can tell between those dots. So that's called the resolution limit. Yeah.
This isn't something as much as probably beyond the final, but if we know what the PSF is it possible to get the information?
Yes, that's some like super resolution microscope. That's one type of super resolution. So you can deconvolution, then figure out what is the center of that sample. Yeah, you can do that. Okay, so then this is a super simplified structure of a microscope. So remember previously you need one excitation and you also need to collect those emission. So here shows you if you have an excitation beam from this side, then this mirror is called dichroic mirror. So this mirror, the special thing about this mirror is it reflects some wavelengths of light and pass through another wavelength. So in this case, if you are exciting your sample with this purple light, then it will be reflected and into the objective, then excite the sample on your sample plane. Okay, then the second process, emission. So you need to collect whatever the light is emitting from your sample. Then this light actually go back through the same objective. Then you can pass through this dichroic mirror, then reach your detector here. Okay, so basically two process, one excitation, one emission. Okay, so these are some more figures showing you these two major type of microscope that people are using, especially in vivo functional imaging. I don't think you need to understand every part of this, but let's just briefly go through. So this confocal microscope is you have a laser light coming from this way, then upward, then you see this same dichroic mirror. So this excitation light can pass through this mirror and reach this is a scan mirror. So this scan mirror can change the beam directions, and a different beam direction result in a different point on your sample plane. So you can go through every point on your sample. Then what happens after you excite those molecules is so the same emission beam travel from the same optical path, then reach here. So remember, this is a different wavelength. Then after it reach this dichroic mirror, it will be reflected to another pathway. So then you can separate these two, this excitation and emission. So this emitted light go through some lens and reach your detector. Okay, so that's the general structure. And the key structure for confocal microscope is this pinhole. So imagine what you want to do is to excite only one specific layer in your sample. So for example, a cortex has different layers, and you only want to excite some deeper layer, but you don't want any interference from upper layers. So by this pinhole, you can reject any emission light from the upper layers. So if you look at this diagram, so only this is the point you want to excite and collect. And so if you follow this solid line, then it goes pass through this pinhole and reach the detector. But anything above or below this plane, then if you follow this path, then that part of light cannot reach your detector. So this is one technique of, it's called optical sectioning. So you can only image one single plane on your sample. Okay, then another similar idea is called two-photon microscope. So remember, two-photon is you use a different light wavelength, but at the same time you have to make sure those two photons arrive at the same time. So another special design for two-photon is you have to use it's called a very short pulse laser. So you can concentrate the photons both in space and in time. So in this case, if you compare which part of your sample is excited, then two-photon is only your focal point. Only this small point is excited. But for confocal, like the whole cone on the top and bottom are, so everything labeled in red is excited. And also they're emitting light. Okay, so if you compare these two structures, still same idea. Excitation light come from this way, then change the angle, then scan through your sample plane. And so in this case, you don't have to reject those light coming from out of focus part. So you don't need this pinhole here. And what you only need to do is to collect whatever is emitting in your sample. So yeah.
In the two-photon microscope, we moved the tube to collect the samples closer to where the objective actually is. Why couldn't we do that in the confocal microscope?
Do you mean move the tube, move the
Right, so
PMT?
Where we actually see it. So at the very end, we traveled all the way back in order to measure the thing. While in the two-photon microscope, we measure very close to the objective. Why can't we measure alpha very close to the objective in the confocal microscope?
Yeah, that's actually a good question because I didn't mention it in detail. If you really follow the optical path, you will figure out that you have to go through this same scan mirror to reach the same point every time. That's called D-scan. So every time you're changing this red beam, and also you need that green beam to travel like from a different angle than to the same point, right? So, but for two-photon, although you're changing this excitation beam, but because every time you're only exciting one point, and you just collect whatever you get from that one point. Yeah. Okay, any more questions? I guess that's all for my lecture. And what else? Okay, then I'll probably quickly go through some of the stuff before exam 2. So my suggestion is maybe start from those 4 core lectures, Dr. Wang, Dr. Zhang, and Shamir and Winslow, and especially those parts that are not tested in previous 2 exams. And for the guest lectures, maybe Dr. Kirkwood and Dr. Connor, they gave 2 lectures, I guess. And Chip will not be on the exam. Maybe FRMI, I'm not sure, but the neuroeconomics part is definitely will not be on the exam. And Shram and Sarma just get some basic big ideas of their lecture. Okay. Oh, not this slide. Okay, then I'll just give a very big picture stuff overview of what we have learned throughout this 39 lectures, if you count the last one. And so remember from the first lecture, we talked about the CNS and PNS, the general division of the whole nervous system. And you have been repeating all this stuff in the following lectures. So for the central system is key thing to remember is the afferents refers to those coming from PNS to the CNS. And you also learned some of the first stage in CNS that receive the peripheral stimulus. For example, like vestibular cochlear nucleus or vestibular nucleus. So they're all the first stage in the CNS that are receiving inputs. And the efferents are just coming from the CNS to PNS. And the last stage in the CNS is usually the motor neurons. Okay, then for the peripheral part, what I can think of is the major 4 types of somatosensory receptors. And for auditory system is the hair cells, and also hair cells are also in the vestibular system, so in the semicircular canals, right? And for visual system, I didn't classify visual system into the peripheral part because retina is technically part of CNS. Okay. Question. Some more big pictures. A neuron, a structure of a neuron. Some important thing to remember: a neuron has multiple dendrites, and those dendrites have spines, which are the little protruding part that receiving some other neurons' inputs. And you have an axon, usually only 1 axon for each neuron. And there's a spike initiation zone, which is the initial segment of that axon, where you see the first action potential. And also for axon, you have the myelin, and those myelins are formed with some other type of cell, which is glial cell or Schwann cell. And you have soma, which is the cell body. And so some different types of electrical signals. So the action potential, you already know this phenomenon called all or none. And also they have refractory period. So another type of electrical signal is the membrane potential. So this refers to the potential inside the cell relative to the potential outside of the cell. So to describe this membrane potential, you should only use depolarization or hyperpolarization. But you can use these 2 words to describe action potential. Because in the exam, I think exam 2, I saw some answers like you can say a neuron becomes, so if you say a neuron becomes more activated, that means the firing rate, the action potential rate becomes higher. Then if you say depolarized, that means the membrane potential becomes higher. It doesn't necessarily generate any action potential. Okay, then EPSP and IPSP is just still also membrane potential, but more specifically refers to those at the synapse. Okay, so E refers to excitatory and I refers to inhibitory. Okay, then you have the chemical signal, which are the signal if you want to transmit information between neurons. And first step is there are some vesicles in the presynaptic terminal. These are just some membrane balls, and you have neurotransmitter inside of those vesicles. So some of the major type of neurotransmitters we have been talking about is glutamate, which is a excitatory neurotransmitter, and acetylcholine, which you have heard about in the first Schramm's lecture. So it's usually used in the neuromuscular junction. And this GABA appears in Dr. Connor's lecture when you are talking about the horizontal cells, and those cells are inhibiting those nearby bipolar cells, and those inhibition is using this GABA neurotransmitter. So this GABA is an inhibitory neurotransmitter. Okay, then in order to receive those informations, then you need receptor on the postsynaptic cell. And these are the two key receptors we have talked about. So these are both glutamate receptors. So they both receive glutamate. And NMDA especially is important because it allows calcium to flow into the cell and generate LTP or LTD. Right? And what's special about AMPA is once LTP happens, then you get those more AMPA inserted into the postsynaptic terminal. Okay. Okay, then just some important ions that sodium and potassiums are important for action potential. And remember, in the hair cells, the endolymph has a lot of potassiums. So those are for the depolarization of hair cells. And calcium is important for LTP and LTD. Also, I have mentioned before, if you have an action potential, then you open those calcium channels, then you can see the calcium influx, which allows you to do calcium imaging, functional imaging of a cell.
Yeah. What does the calcium get up to again? It has something to do with the glutamate channel, right?
Yeah. So LTP is caused by opening an NMDA receptor. An NMDA receptor is a glutamate receptor.
Okay, so you get an action potential, calcium comes in, and that activates the NMDA? When I talk about NMDA, I'm talking about at the synapse part or at the dendrite. And so in this case, action potential usually generated at the axon or somewhere near the soma. Right? I forget how calcium works in LTD.
Okay, LTD, you need to open an NMDA channel, and you need two conditions. One is depolarization, one is glutamate. And remember, depolarization describes the membrane potential change at that synapse. Then once that is opened, then you can trigger more AMPA receptor insert into that synapse.
What is calcium doing?
Calcium triggers that pathway, which allows more AMPA receptors inserted. Remember, there's a more complicated process that you can go back and check.
Think that will be on the exam?
I'm not sure. Okay, then what else? And some math you have done before. So the first bullet point is what I want to clarify is, remember in the first exam you're asked to describe what is a Poisson process, and make sure you understand what is a stochastic process and what is a probability distribution. So in terms of spike arrival, that event can be described as a stochastic process. But if you calculate the inter-spike intervals, those intervals are exponential distribution. So you can mix these two concepts. One is process, one is distribution. Okay, and some other stuff. So PDF point density function and probability distribution function, or cumulative density function, CDF. And so this can be expressed in either little p or capital P. And also in Dr. Shadmir's lecture, he used this PR, which stands for the probability of something. So you can describe probability of a random variable, capital X, is smaller or equal to this little x as P, capital P of X. Okay, just to make sure you understand all these notations. So this capital X underneath means the name of that random variable. And this little x inside of this bracket is the value of that random variable. Okay, so you can put anything like x less or equal to x into those capital P's. Okay, and also maximum likelihood estimation is to find a stimulus or to find x which give you the maximum probability of seeing some response. Okay, just to clear those concepts. Okay, and what else? General questions can you think of? Okay, I'll maybe jump to some comparison between those three major sensory systems we have talked about. So first of all, the somatosensory system, you get inputs from actually the whole body, and those inputs come into the spinal cord from dorsal root ganglia cell here, and it enters the body from the dorsal part and travel along the spinal cord, then all the way up to here. The green structure is thalamus, then relays to the somatosensory cortex. Okay, and if you consider auditory system, then it doesn't go to the spinal cord. It only start from the cochlea, then go into the first stage in central nervous system is called cochlear nucleus. Then the next stage is MSO, LSO, which receives bilateral inputs. And if you go further up, then there's structure called IC, inferior colliculus. And the last stage before cortex is called MGB, which is a part of thalamus. So then if you compare visual system, you're just directly going from retina to this thalamus part. And the visual part in thalamus is called LGN, right? Then so it's a much shorter pathway compared to auditory system. And after this, both of them are reaching cortex regions. So for visual system, it reach V1, and auditory cortex, it reach primary auditory cortex, A1. Okay, cool. And what else? Okay, there are just some more big pictures. Remember these areas we're talking about? So auditory here, visual here, and motor is pre this central sulcus, and somatosensory is after, like post central sulcus areas. And these two specialized areas, one is Broca, which is important for speaking, the production of speech, and Wernicke is more responsible for understanding speech. So you see that production part is closer to this motor area. So everything like in the front of this part is more related to motor. Okay. And what else? Just some more general big pictures of where things are located. So remember, Dr. Kirkwood and Dr. Zhang has talked about the hippocampus, which are a structure shown here in the purple. And Dr. Zhang also talked some other areas like entorhinal cortex and something. So those are all structured inside somewhere around here, so in the middle of your brain. And so for the eye movement and control, or saccade control circuits, those happens in the brainstem. So these are slides from Dr. Shadmir's, and this is a figure show. I think this is from the book chapter that Dr. Cullen gives you. So these are the structures inside of this brainstem. And what else? Okay, then this part, thalamus, is where every sensory system go into thalamus, then project to the cortex. And spinal cord, just some memorizations for segments, and each segments control different part of your body. And this is a flipped structure, so this part is dorsal and this part is ventral. So dorsal is where sensory signal goes in, and ventral is where you output the motor commands. Okay, I don't think I need to go through all this memorization. These are just slides from the first review session. You can go through it by yourself. And I guess I'll maybe, okay, summarize of Dr. Wang's lecture. These are the first several lectures. So first thing is psychophysics. Remember what is a psychometric curve, and differentiate between a detection task and a discrimination task. So I think I need to remind you about these three, actually, three types of different tasks, because we made a correction after exam 1, because Dr. Wang is kind of confusing these two types of experiments. So detection is very easy to understand. You just so the examples are, do you see something, or do you detect something? And discrimination could be designed in two different ways. So one is, if you ask somebody, do you think they are different or the same? That's essentially still a detection task. So you're detecting a difference. So in this case, the chance level is still 0%, which means if you can't tell the difference, then you just tell them, no, I think they are the same. Then, so in this case, the lowest performance is 0%. And another way to ask this question is, so if you give you two stimulus, and I ask you to choose the one which is stronger. So which square is darker, or which sound is stronger. So in this case, if you really cannot detect the difference, then you have to make a choice. Then in that case, the minimum chance level is 50%. So depending on what question you're asking in the task, you should see different chance levels. And so this is a figure that Dr. Wang showed in the lecture. Detection task, very simple, from 0 to 100%, and you take the 50% as threshold. And this actually shows you a 2AFC, which asks you have to choose one that is stronger or weaker. Then in this case, the percent correct goes from 50% to 100%, and the threshold should be 75 correct. Cool. Okay, then what else? So then know how to design a psychophysical experiment. Maybe some of the key words is, first you need to determine what is the range you want to put in the experiment. And you have to make all the stimulus in a random order. And then ask the subject to give you a response. Then each stimulus has to be presented with multiple times. So then you can calculate what is the percentage of correct responses. So those are some key points keywords if you are asked to design an experiment of either detecting or discriminating something. Okay. Okay, what else? Question? No? Then he mentioned, okay, signal detection theory, the 4 types of response: false alarm and hate and false alarm, miss and correct rejection. Right? And you can plot those ROC curves. You can recall by yourself by looking at the homework. And what else? Question about that? No? And receptive field, we have mentioned this multiple times in the later lectures. Okay, I don't need to repeat all this again. And spectrum analysis. Okay, just to refresh your memory, these are the 2 ways to quantify a spectrum. The simplest is to just calculate the mean firing rate. Basically, you count how many spikes across all the trials and divide it by number of trials and the length of time. So that's just give you one number. Right? And second one is called PSTH. And you need to make sure you're clear what should be the x-axis and the y-axis for all these histograms. So for PSTH, the y-axis should be number of spikes per second or millisecond, and the time axis, the x-axis, is just the time. Okay. What else? And okay, now let's compare those two, period histogram and PSTH. So period histogram is actually similar to PSTH, and the difference is you are taking, instead of the whole trial of your spectrum, you're taking multiple integers of the period of your stimulus. So if I say you're presenting a sinusoidal sound wave which has a period of 10 milliseconds, then the whole length of your x-axis should be multiple integers, like either 10 or 20 or 30 milliseconds. So you're dividing the whole trial into different segments, and each segment is n times period. Okay, then if you have phase lock in your response, what you are expecting to see is a bump or a peak in your period histogram. So if you're taking one period, then you should only see one peak. If you take 2 periods, then you should see 2 peaks in the response. Okay. Cool. Okay, then another histogram is ISI. So in this case, x-axis should be still time, but it's a different time. It's not the time we're living in. It's inter-spike intervals. So sometimes people label x-axis as time. It's still correct, but not actually very correct. So you should better label as like ISI or inter-spike interval. And the y-axis now becomes how many intervals you're counting. Okay. Then some features for ISI is if you have a perfect Poisson process, then ISI should be an exponential distribution. But in real case, you don't have this exponential distribution because the spikes have refractory period, so it roughly looks like a gamma distribution. So you have increase, rapid increase, then decay. So that's a gamma distribution. What if you see phase lock in your spikes, then reflected in this ISI histogram, it should be a bump somewhere at the phase locked period. So you still have a gamma distribution, then you see a small bump somewhere else. Okay, questions? All right. Yeah, I think that's all for Dr. Wang's part. And move on to Dr. Winslow. All right. Okay, so 2 sections. One is cable equation. You have done this in the homework and also the first exam. I don't think you have to remember the detailed equations like Vx over V0 equals a bunch of stuff. But if you still have the memory capacity, just store it in your memory before the exam. But the key point is to know what are each parameters are. For example, this Ri means the resistor along the axial direction, and Rm means the membrane, the resistor along the across the membrane, and Cm is a capacitor across the membrane. And you also have this electrical length, which are the actual length divided by a length constant, and also this capital T is the actual time divided by a time constant. Okay, and there are some another intrinsic parameter is called G infinity, which if you take a infinitely long cable, then you can calculate what is the conductance. And this conductance depends only on these intrinsic parameters, like these R's and C's. Okay. And for a finite cable, this is what you have done in the homework and exam 1. So just maybe remember what you need to do in order to calculate something. For example, if you want to know what is the relationship between this Vx over V0, then you need to know what is the length of your cable and what is the G out, which is the conductance connecting to the cable, and also what is the G infinity for that cable, which is only dependent on intrinsic parameters. Okay. Okay, then there are some special case for sealed end. The G out is 0, so you have a much simpler expression for G in and voltage relationship. Okay. Anyway, okay. So the second part is the compartmental model. Still, you don't have to remember all the big matrix of what each part represents, but the general idea is you can divide divide all the dendrite into different parts, and the whole thing can be described as a first order differential equation. Let me go back. Okay, so this is the whole dendrite. Then you can divide it, model the dendrite with this, and each of this Z is the impedance across the membrane. So it's like a parallel resistor and capacitor. Okay, then this V is actually a vector along those dendrite. So you can align all those V's at each point into a vector, then describe those V with this differential equation. So this is the derivative of V equals a matrix times V plus another matrix times U. So this U is the injecting current at each node. Okay. All these details don't have to remember, but maybe just to understand that each of these A and B matrix represents the parameter of that dendrite. So like how long or how wide the dendrite is encoded in this matrix. Good. Cool. And so what you can do with this model is you actually only cares about the steady state. So you can just allow this V dot equals 0, then you can calculate some steady state, which is V is, you can describe the steady state as V equals a matrix times another vector. And this vector is the injecting current at each node. And so the elements now in this matrix is called Kij. If you still remember, it's the transfer resistance. And so each Kij means if you're measuring the voltage at the node J and you're injecting current at the node I, then Vj over I equals Kij. Right. Also, by the same token, you can also let Kii, which is Vi over Ii, which is just you're observing the voltage and also injecting the current at the same point. then that KII is just the input conductance to the cylinder. Right.
Would that be the resistance? Conductance?
Resistance. Sorry. Yeah. Okay. Then this is just to show you what does this transfer resistance mean. And if you're only observing the steady state, then the whole network just becomes a large resistor network. And everything can be described by this transfer resistance. Okay. All right. Then he also mentioned some phenomena. Okay, so this is this. What else you can do with this model is to model a synaptic input. And the way to model a synaptic input is consider the synaptic input as a conductance plus a battery. And this battery refers to the reversal potential of that synapse. So if you have an extra synapse input here, then it's basically just inserting this part of circuit into this part. And now you can calculate this current injected to that node. Then also you can, by the transfer resistance, you can calculate what is the effect of that current to the other parts of the dendrite. So that's Vj can be expressed as II times Kij. Right. So there might be some calculations, but they'll be like pretty simple calculations, just like this. Okay, then there's a phenomenon called synaptic saturation. So consider this circuit, because the maximum voltage change you can cause at that node is limited by this battery, which is E, right? So the maximum, this gives you the maximum voltage change you can see at a soma, and you are injecting current at some point I. So this maximum voltage change can be expressed as a function of E, which is the synaptic reversal potential. So there's a limit for how large you can cause by a synaptic input, how large voltage change you can cause. And he also mentioned if you distribute those snaps, then you can avoid those kind of synaptic saturation phenomena. Right? Okay, what else did we talk about? And we talk about a single neuron can be, a single neuron can do any logical computations. So we mentioned the AND gate, which you have done in the homework. So you have to have both of the synapses turn on together, then cause some voltage change, then action potential generation here. And another is the AND NOT gate. So it involves one excitatory synaptic input and another inhibitory synaptic input. So if you consider this part is soma, then if you turn on this, and if you also turn on this, that means this injectant current will be like leaked from this input, because you have another inhibitory input here, right? So this is called shunting inhibition. And in order to generate some action potential here, you have to keep this off. So that means you have on here and off here. Then you can turn on this part, which is soma. So that's the basic idea of an AND NOT gate. So you have to have this, but not this. Okay, cool. And this is another example he gave in the slides. So this is actually a retinal ganglion cell, and if you excite those, each of these are another cell, another neuron. And if you excite those neurons sequentially from the left to right, then if you follow those synaptic input, then you can see this is excitatory and this is inhibitory, and this is excitatory and inhibitory. So if you excite this one first, then it applies an inhibitory input right here. And at the next time point, if you excite this one, then the inhibitory effect is still there. So even if you excite this one, then this one will be shunted by this inhibitor input. So if you excite all those cells from left to right, then this cell will not fire. So this is called a null direction. And for this cell, the preferred direction should be the opposite. So if you excite those cells first, then sequentially from right to left, then you can avoid those inhibitory effects imposed by the previous cell. Okay. Yeah, you can look at the circuits in detail, but the general idea is you can have this in the, it's called from the excitatory synaptic input to the cell. You can't have any inhibitory inputs. Okay, I think that's some big pictures for Dr. Winslow's part, and this is a summary slide in his lecture. You can just briefly go through by yourself. Again, don't have to remember any complicated equations. And you won't need any calculator in your exam. So, all right. I guess I will stop here. If you have more questions, you can come to me and talk. And I'll upload those slides. All right, what else?
So you said we don't need to remember any like complicated formulas, but like on Winslow's, I think last time we had to like memorize those like formulas for a sealed end or like.
Yeah, those I don't think you need to remember again. Anything involves like cosh or tangent of hyperbolic functions. Those. Tomorrow is Tuesday. Office hours is on Wednesday.
Okay.
Yeah, but I'll be in medical campus tomorrow.
Okay.
So
can I just be emailing you?
Yeah, sure.
Thank you.
Or post on Piazza.
Okay.
Yeah.
I have a question about the short pulse laser in particular.
Okay.
You said that it's because the 2-photon microscope needs to have 2 different wavelengths fired at the same time.
Now 2 different wavelengths. So still 2 same light wavelengths, but longer wavelengths.
Wait, how can it be the same light if it's at 2 different wavelengths? I don't understand that part.
Okay, so 2-photon is, remember, compared to 1-photon is you excite with a blue light, which is a short wavelength or higher energy wavelength. And 2-photon, you are using 2 photons with the same wavelength.
2 photons with the same.
Yeah.
Okay, that's why it's called the 2-photon.
Yeah.
Okay, so
each photon has a lower energy, but if you sum up, it's still the high energy.
Yeah. So does the short pulse laser just ensure that they hit the same type of electron at the same time?
Yeah.
And make sure that it makes a jump?
Mm-hmm.
Okay.
Yeah,
that's what I was unclear.
Yeah.
Thank
So
you.
you have to be very coincident for those 2 photons.
Yeah.
And that way you get rid of the noise, right? You only focus on one area with the 2 photons?
So everything else in the cone is not excited.
If it was
only
confocal, then it would kind of excite everything around it.
Yeah.
And emit light, that's why you need the pinhole.
Yeah.
The only thing is you get rid of the pinhole, right? You don't need the pinhole anymore if you do 2 photons.
But like, if they want to.
And you need a much more expensive laser.
To do 2 photon or confocal?
to 2 photon. Just that short pulse
But then
laser.
is that better then? Like,
Of
don't you get
course.
the same thing by using a pinhole?
At like a fixed.
Or is it still
Actually,
bad resolution?
It's higher resolution. Actually, the resolution should be comparable, not very much improved. But the key point is, I didn't mention in the lecture, because light is not invasive actually. So if you excite everything, especially in a living animal in the brain, then actually cause some damage. With photo bleach, photo damage. So you don't want to excite everything.
Oh, okay.
In the
And then you're using confocal and 2 photon because the light won't. You don't have a light source underneath it.
Yeah.
Basically.
Mm-hmm. Yeah.
Okay, so I'm still a little confused about the hop field, like Zhang's stuff. In terms of, well, I mean, I didn't look at it yet, but maybe that's why. But it's really hard to listen to his lectures because I always fall asleep.
Yeah.
Like, oh man, it's just not practical. It feels like to actually watch it. At least this part. Like his whole head orientation movement, that's fine. It's like conceptual, but like so.
Andy did a good job of. If you're having trouble with
Yeah.
his lectures, listen to Andy's like again for the
For this review, did it work?
Yeah.
Like you guys were like fumbling around, so I was like, wait, does something not record?
Did I? Oh, because the memory card was full.
Oh?
I
Yeah.
don't
He
know
went
how
first,
much.
so.
Yeah, I think his part is captured.
Okay.
I'm not sure, but I recorded with my phone. Oh, it's still recording.
Yeah.
You're on tape for falling asleep.
I'll upload.
-2.
Okay, I'll take a look at it again. But is it possible to come by? Like, do you guys have office hours? Or will you
On
have-
Wednesday.
On Wednesday?
Mm-hmm.
Do you think we can look at our past exams?
Yeah.
Look it over again, kind of.
Are you gonna?
Yeah, just
To
look-
see the mirror mistakes and stuff.
Yeah. Do you think that's possible? I mean, it's a lot of tests.
Probably not for a regrade at this point.
Well,
but
not for
you could-
regrade, but like just to review, kind of.
Yeah, I have them in Clark.
Okay.
Actually, you can stop by now if you have time. This afternoon.
Is it okay to like come before the office hours? Like 30 minutes before to look at it?
Oh, on Wednesday? Yeah.
Yeah, okay.
Andy scared him when he talked about the reinforcement learning in the context of having to see yours.
Yeah.
Like, sucks to be you, you didn't see it.
Now you have to do supervised learning. Otherwise, you have to be stochastic for final.
Yishi, I had a question about 2 photon radiation. So is there no PSF? Or, like, is there width in the PSF of 2 photon imaging?
Yeah, everything has PSF.
Okay, so what's the source of that for 2 photon? Is there
It's still as long as you have the objective. So as long as the light passes through the objective and it's being focused, and that point is not extremely small. So it's limited by the objective.
Okay, and the diffraction related stuff?
Yeah. Mm-hmm.
Okay, but the reason why like the cells that you excite is so small in 2 photon, is it because it's just 2 photons that are being used, or what?
Yeah, I mean by small I just mean maybe close to the PSF size.
Okay.
So that's small enough.
Okay.
Like maybe 1 micron.
Okay.
Yeah.
Okay, got it.
Yeah.
Alright, okay, thanks,
Yeah,
Yishi.
no problem. Good luck.
Thank you. Appreciate all your help.
Problem.
All the best with your research work and everything.
Thank you.
Yeah.